Today we will talk about lamp, loop mediated isothermal amplification. This is a isothermal. This technique is performed at 60 to 65 degrees Celsius under under controlled temperature. Lamp technique was first reported in 2000 by Naomi and its co-worker in Japan. It amplifies the target segment under isothermal condition, and the loop structure is formed to amplify the target DNA sequence. Lamp was created to to lamp was created to lamp was created to overcome the drawback of PCR technique. Target The target DNA from pathogen is amplified by the use of VST DNA polymerase, which is isolated from Bacillus stereothermophilus. The lamp consists of 4 to 6 design primer which hybridizes into 6 to 8 different parts of target sequence. The type of primers which are required are forward inner primer, forward outer primer, backward inner primer, and backward outer primer, forward loop primer, and backward loop primer. First, the DNA of interest is identified with 3 target regions and the 3 backward target regions. Primers are designed according to the target sequence based on conserved region. Then, based on each sequence of 6 target region, lamp primers are designed. Now, denaturation of DNA containing target is done in two separate complementary strands. After that, forward inner primer binds to F2C region and primes the polymerase by adding complementary nitrogen bases into 3 to 5 direction. Similarly, the forward outer primer F3 binds to the region upstream to from the FIP binding site. Therefore, the new strand form created by the action of forward inner primer and which is now unzipped by forward outer primer as it synthesizes the complementary DNA. Similarly, backward inner primer binds to newly made complementary strand at the B2C region, synthesizing the complementary DNA in 3 to 5 direction. Now the backward outer primer F3 binds upstream from the binding site of BIP. B3, which is backward outer primer, now unzips the new strand which was formed by the action of BIP as it synthesizes the complementary sequence. Finally, a new synthesized strand contains two complementary regions, F1 and F1C, B1 and B1C, which makes the dumbbell structure with two loops, which we can see how forward internal primer attached to the strand, and then backward internal primer this form the complementary strand and which is then converted into loop. Uh, Loop structure forming is the basic of lamp autocycling process. forward inner primer and backward inner primer bind an F2C and B2C region respectively. Self-priming of F1, F1C and B1, B2C occurs and in continuous cycling resulting in further round of synthesis. In addition, forward and backward loop primers provide other priming site to increase the amplification rate. The binding site of LF is a sequence between F12 and F1 and F2 region of the target, whereas that for LB is a sequence between B1 and B2 target region. LAMP is detected by many methods like gel electrophoresis, fluorescence, chemiluminescence. In this we can see that there are ladder-like structure formed. LAMP has many advantages like it is highly sensitive technique and uh, it can detect the at the real time. This technique is rapid which takes around 60 minutes. It uses different polymerases which have high strand displacement activity. It has simple protocol without any complicated procedure, and it has different preparatory steps as in many PCR systems. It requires low cost equipment and the entire process is run under constant temperature. LAMP technique is used for the detection of many foodborne diseases caused by Salmonella, Legionella, Listeria, norovirus, etc. Detection of food pathogen using LAMP technique uh, ensure the food safety and quality. This technique is also used to detect the corona and influenza viruses, and this is a rapid technique. HIV, Vibrio cholerae, and other pathogens are also detected using LAMP technique. Like any other technique, LAMP also has some drawbacks or limitations. Sometimes it can be difficult to design the LAMP primers because of its high sensitivity. Some sometimes it can give the false positive results, which can occur because of the cross contamination. Mainly because there are if there is many if there is any air remaining in the assay, sometimes it can be difficult to quantify the results. Future perspective, lamp can be used to detect the viruses or diseases which are neglected or are present in tropical areas, and to detect the plant and animal diseases. Uh, this technique uh, is this technique can be performed in limited environment and in limited equipment. So therefore the research are going on to how to advance the technique.